Hello, my name is Fan Lo. I'm going to give you recorded lectures on cell membrane and transport processes. I hope you like the lecture, and we will book some time later for you to ask questions. We would like to achieve two things after the 2 hours lecture, and this will be broken down into 3 parts. So first of all, we would like to describe the structure of plasma membrane and the importance of membrane transport. The second one is we should be able to describe different types of membrane transport, including their energy requirement. So these different types are simple diffusion and how to quantify this, facilitated diffusion, primary and secondary active transport, endocytosis and exocytosis, water transport and osmosis. So first of all, why are we studying membrane transport? How important they are? As we know that the cell membrane is a boundary of a cell, and the substances within the cell will have different concentration from the substances outside of the cell. And for the metabolic substrates like glucose and amino acids, you would have higher concentration within the cell. But for the metabolic end products like the waste and toxic material, for example carbon dioxide, you would like to move out of the cell, so that is to exclude them out of the cell. And we also know that many enzymes, they have to work within a range of their pH value, and they have to the cell have to maintain the correct ionic concentration. So cell membrane transport is very important to maintain the pH and ionic concentration. Cell membrane is also very important in the regulation of cell volume, because these cells, they are soft and flexible. It's not like plant cells can have a rigid shape. So the cell volume is dependent on the membrane transport. Finally, it is important for the generation of ion gradients for signaling function. So what do we mean by that? That is, when action potential is produced, that is because different ions have different concentration within the cell and outside the cell. And after the action potential, the ions have been redistributed, and how to restore the pre-action potential status, and that is a job for the membrane transport to do. So membrane transport is very important in maintaining the cell function in various ways, and that is why we need to study membrane transport. So this is a model of cell membrane. If we look at the main structure of a fluid mosaic model, are the bilayers of phospholipid cells in the this majority part. We say that it is a fluid mosaic model because it is very well organized like a mosaic, but it can be flexible with certain flexibility, so that is why it's called a fluid mosaic model. On this model, we can see that there are phospholipid bilayers and protein which can be integrated or peripheral, and we also have carbohydrates. So this is extracellular fluid, and here is cytosol, that is the intracellular fluid. So if we look at the words to explain them, there are bilayer of phospholipids with hydrophilic on the sides, on both sides, and then hydrophobic between the bilayer. Membrane proteins can be integrated, can be peripheral. Membrane carbohydrates, it can be combined with lipids, with glucolipids, or combined with protein, become glucoproteins. These are very important for cells to identify substance from outside. So let's look at how does a cell membrane look like from the building blocks. So first of all, we start with a phospholipid molecule. This is hydrophilic side, and the rods are hydrophobic side. And now, if we have many of those, they have arranged very nicely to form the polar heads outside, and then in between, it is the non-polar heads formed the hydrophobic region. And we have extracellular fluid, and intracellular fluid. We also have integrated proteins and peripheral protein. So that is the analysis of a cell membrane. Now let's look at the distribution of molecules across the cell membrane. This example comes from a rat skeletal muscle cell. So on the left column listed the components, that these are the different molecules. The middle column is intracellular concentration in millimolar, and the right side column is the extracellular concentration, also in millimolar. So we have highlighted two types of ions. One is sodium with the intracellular concentration 16 millimolar, extracellular concentration 150 millimolar. So it's about 10 times higher outside the cell than inside the cell. On the other hand, potassium ions have more than 20 times higher concentration within the cell, 152, against the outside the cell is 6.4 millimolar. And why we highlighted these two types of ions? Because these are the crucial ones for action potential to be produced. You're going to learn action potential in semester B. And here, when action potential happens, the sodium channels open and sodium ions rush into the cell from higher concentration to the lower concentration. And after the depolarization, during the hypopolarization, potassium ion would go from intracellular to extracellular when the potassium channels are opened. So these are some particular example for membrane transport. And if we look at the chloride, this chloride has a similar concentration folder with the sodium because they are going together. And calcium is an interesting one. Outside the cell, it has about 2 millimolar, and within the cell, the concentration is very, very low. It's below 1 times 10 to the minus 3 millimolar, and which is usually 10,000 times lower within the cell than extracellular. And that is why calcium is a very important second messenger, because the huge gradient in concentration within the cell and outside the cell. So when the calcium channel opens, some ions just rush into the cell, and these ions will be enough to trigger some intracellular process like release of hormones and muscle contraction, so release of neurotransmitter like acetylcholine. So calcium is a very important second messenger. And after the action potential, after muscle contraction, the ions need to be restored to their resting status, and that is where membrane transport is very important, that they have the function to maintain those ionic concentration. Now, if we move down to look at the creatine phosphate and ATP, they have zero concentration outside the cell and a very high concentration within the cell, and that is because these are the energy sources for muscle contraction and for many other cell processes. And sugar is an important metabolic substrate. So within the cell you have a high concentration of phosphate sugar with other form, but glucose is zero. Extracellular, you have 5 millimolar glucose. The reason is it is energy saving to move from higher concentration to lower concentration, and as long as they come into the cell, they have been transferred to another form, so the glucose concentration maintained at zero to ensure that the movement from outside the cell to into the cell is energy free, not requiring any energy. We will talk about this later. And amino acids has much higher concentration within the cell. That is because amino acids is not only for the building blocks for protein, for enzyme, and for many other hormones. Amino acids is also used as neurotransmitter. Urea has the same concentration within the cell and outside the cell. Protein has much higher concentration. So this table illustrates the distribution of different molecules across the membrane, and that provides some information for us to understand the membrane transport. Now, a brief outline of membrane transport. We have listed the different types of membrane transport. Here, it's been grouped together into one is a passive transport, that is no energy required. Within that group, we have simple diffusion and facilitated diffusion. Within facilitated diffusion, we can also divide them into channels and carriers. The other group is active transport, which is energy required. For active transport, you have the first type, primary type of active transport, secondary type of active transport. You can also have transport via compartments, that is endocytosis and exocytosis. So now let's look at the simple diffusion and also how to quantify this. Remember that simple diffusion goes through the lipid bilayer directly. So here is an illustration of simple diffusion, so which is increase disorder and provide energy. So it does not require energy to do that. So on the left side, we start with initial state. Here we have two compartments. O represents outside the cell. I represents inside the cell. In between, you have a dashed line representing the cell membrane. The start point is on the left side, which is outside the cell. a certain type of molecules have much higher concentration than the right side, which is inside the cell, that is the initial state. When after certain time it reached the final state, as you can see that in the final state the two sides have the same concentration. So what has happened? Let's look at the animation here. With simple diffusion, it is increasing the disorder. So this is the start point. You can see the left side has much higher concentration, and after this movement, both sides have got the similar concentration. So the reason behind this is the thermal motion of the molecules. It can move both sides, but the net movement is from higher concentration to lower concentration. So if we look at this diagram, the time 0, which is the initial state, for example, if we start everything with one side, so the other side is with concentration of 0, after certain time, one side is increased the concentration, the other side has decreased the concentration. Eventually, the two sides would reach the same concentration, that is the final state. So concentration can be expressed in molar or millimolar, and the difference in concentration is called delta C. So that delta means the difference, and this difference we also call it concentration gradient, and that is a very important concept in membrane transport. So if we describe the simple diffusion, it goes through the lipid bilayer directly. No energy is required, and that is the result. So the driving force for the simple diffusion is random thermal motion. So with the thermal energy, then the molecules are jumping around all the time, and the direction of the transport is determined by the electrical chemical gradient. So what do we mean that? Electrical means whether it's negatively or positively charged. Chemical means their concentration difference. So electrical chemical gradients means it would move from higher concentration to lower concentration, and the negatively charged molecules, particles, are likely to move towards the positively charged side. For example, sodium ions is positively charged. It could intend to move towards the negative potential area. So the last point describe the rate of transport. So the definition of rate of transport is number of molecules cross the membrane in a given time. And this rate is dependent on the driving force, which is how much concentration gradient, how different the concentration gradient is across the membrane. Second one is how big the membrane area is, and then how thick the membrane is, and what about the permeability. So let's look at these four factors one by one. First of all is the simple diffusion, how the concentration gradient is affecting the transport rate. We start with 0. If two sides have the same concentration, then the net flux, which is the net movement across the membrane, is 0. That means you have already reached the final state. You don't need to move around again. And when the concentration gradient increases, there is a linear relationship between the rate of transport to the concentration gradient. So the more the concentration gradient is, then the quicker the movement would be. So that is how to quantify the simple diffusion. The next one is area. So if you have that big membrane, then certain number of molecules can cross this area. If you quadruple the area for the movement, then the rate of movement is also quadrupled. So that is the relation. The bigger the area is, then the higher the speed of movement would be. And this is easy to understand. For example, if you are crossing a river by swimming, and the longer the river bank is, then the more people can swim at the same time. So more people would be able to cross the river at the same time. And the next thing is permeability. So what do we mean by permeability? That is dependent on the individual molecule, how easily they can cross the membrane. So if the lower permeability with a certain concentration gradient, the movement speed is low, and if it is a higher permeability, then with the same concentration gradient, the speed of movement is much higher. So a person that is fit and trained for swimming can swim much faster than a person like me, just barely can swim. So this permeability is the ability of crossing the membrane with individual molecules. So what kind of factor can determine this ability? So let's look at this one. That is called the membrane permeability. So with a higher permeability, that means easy to cross the membrane, down to the lower permeability that is difficult to cross the membrane. The group that are easy to cross the membrane are the small non-polar components, and the examples are steroids, gases, so carbon dioxide and oxygen. So these are the important gases for respiration, and that is why in respiration the molecules cross the membrane by simple diffusion, because these are the very small nonpolar components, very easy to cross the membrane by simple diffusion, and we say that they have higher permeability. Other molecules are anesthetics, water. Water is small polar components. The water can cross the membrane without problem. That is because the aquaporin channels. We will talk about this in the osmosis. Urea is easy to cross because it's a small nonpolar components. Glycerol is also easy to cross. When the size of the molecules getting bigger, becomes large polar components, then it becomes more and more difficult to cross the membrane by simple diffusion. Ions, although they are very, very small, but they are charged, so they are very difficult to cross membrane because the between membrane hydrophobic area cannot be crossed by ions. So the non-permeable ions molecules are sugars because they are too big. Amino acids, they're not only big and they can also be charged. Nucleosides and ions. So bear this in mind. This can help you to distinguish whether they can cross the membrane by simple diffusion or not. So the membrane permeability is lipid solubility of diffusing substances. For example, oxygen and carbon dioxide, they're very easy to dissolve in lipids. Size and shape of diffusing particle is important. The smaller, the easier it can cross. temperature contributes to the permeability, because the higher the temperature is, the higher the thermokinetics is. So the thermokinetic energy is higher, and then the quicker the movement can be. Thickness of the membrane is inversely related to the rate, because the wider the river is, then the longer it takes you to swim across. So the thicker the membrane is, then the longer it takes to cross the membrane by simple diffusion. So here are some examples of molecules cross the membrane by simple diffusion. First of all are the gases during respiration. Steroid and thyroid hormones with endocrinology, these ones are crossing the membrane by simple diffusion. And also lipid-soluble vitamins can cross the membrane by simple diffusion, which are the A, D, E, and K. So now let's look at the simple diffusion size of the molecules. So on the background, here is the model that we have drawn earlier with bilayer of phospholipid molecules, integrated protein, peripheral protein. Now we have a very small size of molecule. They can cross the membrane very easily. If we have medium size of molecules, they start to cross the membrane slowly, even bigger, even slower. Now, if we have a very big size of molecule, can they cross the membrane? No, it's too big. So this slide illustrates how the size of molecules matter with simple diffusion. Now, let's summarize the simple diffusion with the Fick's law. Fick's law quantifies the simple diffusion. So, first of all is the higher the concentration gradient is, the more the net flux is. So that's the quicker the movement is. Second one says the bigger the membrane area is, the more the net flux is. Then it's the bigger the permeability is, the more the net flux is. So if we use an equation to quantify this, net flux equals permeability times area times difference in the concentration. So this is Fick's law. You need to understand the contribution, which one is proportionally related to the net flux, but you will not be required to calculate how much molecules can be crossed by a membrane within a certain time. So you need to know it qualitatively. Yeah. Okay, that is the simple diffusion. Let's take a break.